right now, in a research lab somewhere out there in the world, there's a mouse living inside a highly sterile enclosure.
Yeah, a very specialized, completely isolated environment.
Right, but, this isn't just your ordinary lab mouse. Growing just beneath its skin or maybe nestled inside its liver is a perfect, living, breathing replica of a human being's cancer.
It's a biological avatar, essentially.
Exactly, an avatar, and it holds the exact molecular secrets to saving that specific human's life.
I mean, I know it sounds like science fiction, but it is very, very real. It's really the ultimate frontier of personalized medicine right now.
It totally is. So, welcome to today's deep dive. We've got a massive stack of sources today, everything from, decades-old surgical logs to some seriously cutting-edge clinical trial data and genomic research papers.
Some really dense material, yeah.
Yeah, super dense. But our mission today is to unpack this fascinating world of patient-derived xenografts, or PDX. Basically, we are looking at how scientists create these living avatars to test potential cures before they are ever, administered to the patient.
Right, and to really understand why we even need these avatars in the first place, we first have to understand the fundamental conundrum of cancer treatment.
Which is what, exactly?
Well, we like to think of medicine as precise, right? You have a bacterial infection, you take a specific antibiotic, and boom, it's gone. But cancer isn't just one single disease. And tumors, they aren't just solid blocks of identical clones.
Right. If you look at the research, tumors are incredibly complex. They're like these evolving, chaotic ecosystems.
Precisely. They exhibit what we call intratumor heterogeneity.
Intratumor heterogeneity. Let's break that down for everyone.
So within a single tumor, you have these distinct subregions. You've got completely diverse cell phenotypes, different genetic mutations, all just mixed together in one mass.
So it's not just one target you're trying to hit?
Exactly. And because of this, traditional cancer drugs often fail. Or, they only benefit a really small proportion of patients because they might only kill off one specific type of cell in that ecosystem.
And historically, we've tried to study these tumors by just growing them as 2D cell lines in a flat petri dish, right?
Right.
Or using genetically engineered mice, or even building those tumor-on-a-chip model.
And we have, yeah. But those methods, they all eventually fall short.
Because they're too simple.
Yeah, a flat layer of cells in a plastic dish completely lacks the true three-dimensional spatial architecture of a real tumor. It doesn't have that chaotic blood vessel distribution.
The scaffolding.
Right, the complex cellular scaffolding, or the clinical consistency of the real thing.
Yeah.
We really want to figure out how to destroy a specific cancer. We need a perfect 3D mirror image of it.
Okay, so if petri dishes and flat cell lines fail, the logical next step is taking the actual living tumor and, keeping it alive in a host. What does that process actually look like on the ground?
At its core, creating a patient-derived xenograft model, a PDX, starts in the operating room.
Yeah.
We take fresh tumor tissue directly from a human patient during surgery or a biopsy.
Right.
And instead of putting it in a jar of formaldehyde or preservative, scientists literally surgically transplant that living tissue into a mouse, keeping the actual 3D tissue structure completely intact.
Wow. And this isn't some brand new, unproven idea, is it? We've been doing this for a while.
Oh, not at all. We can actually trace the foundation of this all the way back to 1969.
1969. That's wild.
Yeah. Two scientists, Rygaard and Paulson, they successfully implanted a human colon adenocarcinoma into what's known as a nude mouse. It was a massive, massive breakthrough.
Okay, wait. Let's think about the biology here for a second, because this is where my mind gets blown. If I take a highly unique seed from an alien planet and plant it into Earth's soil, the Earth's environment is going to treat it as a foreign invader and attack it.
Right, naturally.
So wouldn't a mouse's immune system immediately recognize this human tissue as foreign and just absolutely destroy it?
That is exactly the hurdle. That's the biggest challenge. So to make the biological soil hospitable to the human seed, as you put it, the mice must be highly immunocompromised.
Okay.
We use very specific genetic strains of mice.
Huh.
You read the literature, you'll see acronyms like SCID, NOD, SCID, NSG, or BRG.
Okay, let's translate that alphabet soup for the listeners. What do those acronyms actually mean for the mouse itself?
Right, so they represent escalating levels of immune deficiency. For example, nude mice, the ones they used back in 1969, they lack a thymus gland.
And the thymus does what?
Well, without a thymus, they can't produce T cells, and T cells act as the generals of the immune system. They orchestrate the attacks.
So the mouse has no generals to lead the army.
Exactly. But modern NSG and BRG strains, they take it even further. They're engineered to be severely deficient in T cells, B cells, and natural killer cells.
Natural killer cells being like the immune system's frontline assassins, right?
Yeah, that's a great way to put it, yeah. They patrol the body and just destroy anything that looks foreign or infected. So the rule of thumb in establishing a PDX avatar is pretty simple. The more immunocompromised the mouse, the weaker its defenses, and the higher the success rate of engrafting that human tumor.
Got it. And where exactly do scientists plant this seed? Do they just put it anywhere?
No, there are three main approaches.
Yeah.
Subcutaneously, which just means tucking it right under the skin is the easiest way.
Okay.
Then there's orthotopically, which is amazing. That means implanting the tumor into the exact same organ it came from.
Oh, wow.
Yeah, so a human liver cancer biopsy gets grafted directly into the mouse's liver. It mimics the natural environment. And finally, you have heterotopically.
Heterotopically, what does that mean?
This is when they implant the tumor in highly vascular areas,
Yeah.
places with tons of blood flow, like under the renal capsule of a kidney or, and this is wild, even inside the eye.
Wait, inside the eye, just for the blood flow?
Exactly, to ensure the tissue gets a massive supply of nutrients to kickstart its growth.
Okay, so the human tumor is planted. How long does it take for this avatar to actually become a useful model that we can use?
It can take anywhere from a few days to several months, really depending on the aggressiveness of the cancer. But generally, the tumor stabilizes around 40 to 50 days.
Okay, so a little over a month.
Right. At that point, it reaches about 1 to 2 cubic centimeters. And then researchers perform what's called a passage.
A passage, like passing it along.
Exactly. They carefully remove the tumor, segment it into smaller pieces, and transplant those pieces into a whole new cohort of mice.
Oh, I see.
This multiplies the avatars, giving us this massive testing pool for different drugs. It's like taking cuttings from a plant to grow an entire garden.
Okay, so just to make sure we're tracking the mechanism here, let me recap this first part. We take a fresh human tumor, we preserve its 3D architecture, and we plant it into a mouse whose immune system has been entirely wiped out.
Yes.
We place it where it can get the most blood flow, let it grow for a month or two, and then we divide it up to create a small army of custom avatars.
That's the core of PDX, yes.
I mean, think about it. Imagine you are sitting in a doctor's office facing a really tough diagnosis. How would you feel knowing scientists could grow a custom, living replica of your specific challenge to safely test out the blueprints for a cure before you ever have to take a single pill?
It's profound. It entirely shifts the paradigm of oncology from, reacting to cancer to actually preempting it.
But wait, thinking about this, that brings up a massive logical contradiction.
Oh, I know where you're going with this.
Yeah, if we deliberately wiped out the mouse's immune system so it wouldn't reject the tumor, how on earth are we supposed to test immunotherapy? Because leveraging the body's immune system is arguably the hottest, most promising field in cancer care right now.
You hit the nail on the head. It is the ultimate catch-22 of the PDX model. You absolutely need an absent immune system to grow the tumor in the first place, but you desperately need a highly active immune system to test immunotherapy drugs.
So how do we fix that?
To solve this, scientists do something incredible. They humanize the mouse.
They humanize it, meaning what? They inject human immune cells.
Exactly. Researchers inject human peripheral blood mononuclear cells. They're known as PBMCs in the literature. Or they use human hematopoietic stem cells.
And they put those into the mice.
Right, into these severely immunodeficient mice. Sometimes they even use human umbilical cord blood. These human stem cells literally take root inside the mouse's bone marrow. Wow. And once they take root, they begin generating human T cells, human B cells, and human dendritic cells right there inside the mouse.
That is just mind-blowing. So we are essentially giving a mouse a functional human immune system alongside a human tumor. We are basically building a custom, highly specific human battleground inside a mouse.
That's a perfect analogy. And what's fascinating here is how accurately this miniature battleground replicates what happens in an actual human body.
How so?
Well, when you put the human immune system and the human tumor in the same mouse, they start interacting immediately. The tumor actively educates and exhausts the human immune cells over time.
The tumor educates the immune system, and you mean it fights back?
It does. It's not passive. The cancer cells suppress the immune cells, essentially dialing down their production of pro-inflammatory cytokines.
The cytokines being like the chemical alarms.
Exactly, the chemical alarms the immune system uses to call for backup. The tumor silences those alarms. And through a biological process called trogocytosis, it gets even more sinister.
Trogocytosis. Okay, let's break that down. Are you saying the tumor is physically doing something to the immune cells?
Yes. It's wild. The word comes from the Greek trogo, meaning to nibble.
To nibble, okay.
The cancer cells literally nibble on the human lymphocytes. They rip membrane proteins, specifically immune regulatory proteins, right off the surface of the immune cells and wear them.
Wait. The cancer is literally stripping the armor off the immune cells and putting it on to hide itself.
That's exactly how it works. By stealing those proteins, the tumor disguises itself as part of the immune system.
That is terrifyingly smart.
Right. And because we can observe this happening in the humanized PDX model, the avatar perfectly simulates the exact same immune exhaustion and deception that we see in real-world clinical patients.
Okay, I'm just pausing to visualize this. To test immunotherapies, we inject human stem cells into a mouse to build a human immune system from scratch. Then, we just watch as the tumor and the immune system go to war, with the cancer sometimes literally biting off and stealing proteins from the immune cells to survive.
It's a microscopic war zone.
I mean, imagine the cancer as a malicious hacker actively rewriting the software of your immune defenses. If we can watch this chemical warfare happen in real time inside a mouse, we can figure out exactly which firewall to build to stop it.
Exactly. And if we connect this to the bigger picture, it means we can test checkpoint inhibitors and CAR T-cell therapies in a living system that mimics the exact complex environment of the actual patient.
But of course, no scientific model is perfect, right?
Yeah.
We have to look at the downsides. There have to be some glitches in this biological matrix. What goes wrong inside the avatar?
Yeah, it's not flawless. The biggest issue researchers face is something called stroma replacement.
Stroma replacement.
Yeah, the stroma is the supportive tissue of the tumor. Think of it as the extracellular matrix, the blood vessels, and specialized cells called cancer-associated fibroblasts, or CCAFs.
Okay, so it's the scaffolding that holds the building up.
Right, exactly. Now, when we first transplant the human tumor into the mouse, the human stroma comes with it. But as we passage the tumor from mouse to mouse over time, something happens.
The mouse takes over.
Yep. Usually after 2 to 5 passages, the human stroma is almost entirely replaced by murine or mouse stroma.
Oh, wow. This sounds exactly like the ship of Theseus paradox.
Ha! Yes. That's a great way to think about it.
For anyone who doesn't know, the ship of Theseus is this old philosophical thought experiment. If you replace every single wooden plank on a ship one by one over time until no original wood remains, is it still the same ship?
Right.
So if the tumor scaffolding, its entire support system, becomes made of mouse tissue instead of human tissue, does the drug test still apply to the human?
And that is a major, major debate in the field right now. Because the stroma isn't just dead scaffolding. It actively secretes proteins and influences how the cancer grows and responds to drugs.
Right, it's active.
Exactly. If the stroma is mouse, the communication network changes. The results might be skewed. But there's another flaw, too. The loss of heterogeneity. Remember how we said tumors are a diverse mix of different cell types?
Right, the chaotic, evolving ecosystem.
Well, inside the mouse, it becomes a rapid game of survival of the fittest. Certain subclones of the cancer might thrive and grow much faster in a mouse environment than they ever would in a human.
So the environment selects for different traits.
Exactly. For example, in small cell lung cancer PDX models, researchers found that specific neuroendocrine cells quickly outcompete all the other cell types.
So you lose the diversity.
Yeah, you end up with a tumor that has lost its original diversity.
Yeah.
It no longer perfectly matches the original biopsy. It's a distorted reflection.
And looking at the sources we have, there's one more massive glitch that stood out to me, the lymphoma risk.
Yes, this is a very strange one. So sometimes the patient's tumor sample accidentally includes normal human B lymphocytes that happen to be carrying the Epstein-Barr virus, or EBV.
Which is pretty common, right?
Extremely common. Most adults have had it. But in the highly immunocompromised, totally defenseless environment of the mouse, these EBV-infected B cells can suddenly explode in growth.
Unchecked by an immune system.
Exactly. And that causes a spontaneous aggressive lymphoma in the mouse.
So let me get this straight. You're trying to carefully grow, say, a colon cancer avatar, and suddenly the mouse develops a completely different disease lymphoma, ruining the whole experiment.
Precisely. It completely hijacks the model. To combat this, scientists sometimes have to proactively dose the mice with a drug called rituximab, which suppresses human B cells, just to keep the original experiment on track.
So our biological matrix definitely has some bugs. Over time, the human scaffolding gets replaced by mouse tissue. The tumor can lose its original diversity as aggressive cells take over, and sometimes hitchhiking viruses cause unexpected lymphomas.
The constant balancing act.
I mean, if your custom avatar starts subtly shifting its underlying architecture, how do we adjust our strategies to hit a moving target?
Well, that is where modern technology really comes to the rescue, and it's also how we solve another major problem with mice.
Time. Right, because growing a tumor in a mouse takes weeks or months.
Exactly, and that is time a late-stage cancer patient simply does not have.
Right, speed is everything. So how are researchers supercharging these avatars to beat the clock?
First, they developed a protocol called mini-PDX.
Mini-PDX?
Yeah, which drastically shortens the implantation and drug screening cycle to just a few days. But more radically, they're looking beyond mice entirely. We are now using alternative animal avatars like chick embryos and my personal favorite, zebrafish tumor xenografts, or ZTX.
Zebrafish, like the little striped fish you see in a home aquarium. Why on earth a fish?
Because they reproduce incredibly rapidly, allowing for massive high-throughput testing. That means we can test hundreds of different drugs simultaneously on hundreds of embryos. But the real superpower of the zebrafish embryo is that it is entirely transparent.
Transparent, meaning you can literally look right through them under a microscope.
Yes, it's incredible. Researchers inject the patient's human tumor cells into the transparent fish embryo. But first, they tag the cancer cells with a green fluorescent protein.
So they glow.
Exactly. Using advanced real-time light sheet imaging microscopes, scientists can literally watch the glowing cancer cells spread through the fish's body, attach to vessels, and interact with drugs right before their eyes, in real time.
That is unbelievable. It's like watching a living X-ray.
It really is.
But they aren't just relying on visual data, right? Because the sources mentioned they are merging these avatars with massive amounts of big data and multi-omics.
Absolutely. We aren't just looking at the physical tumor anymore. We are looking at its genome, its epigenome, its transcriptome, and its proteome all at once. For example, scientists are now performing CRISPR-Cas9 dropout screens directly inside the living animal.
Hold on, let's unpack a dropout screen for a second.
Yeah.
How does CRISPR work inside a living avatar?
Okay, so they use CRISPR to systematically knock out or disable thousands of different individual genes across a population of cancer cells inside the mouse.
Like flipping off thousands of different light switches.
Perfect analogy. Then they apply a chemotherapy drug. By analyzing which cancer cells die, meaning they drop out of the population, scientists can pinpoint exactly which genetic vulnerability makes the cancer susceptible to that specific drug.
And they are tracking this at an impossibly small scale now, right? Down to the single cell.
Exactly. A cutting-edge technology called CGET-SEQ allows researchers to map both the genomic DNA sequences and the epigenomic chemical tags of individual single cells at the exact same time. It's a level of resolution we've never had before.
But the barcoding technology is what really blew my mind reading through the research. Can you explain that part?
Yes, the barcoding. Scientists can now inject DNA barcoded nanoparticles into these tumor-bearing mice.
Nanoparticles of barcodes.
Yeah. Each microscopic nanoparticle carries a unique synthetic DNA sequence, a literal biological barcode, along with a specific drug payload. After the treatment, they extract the surviving cancer cells, sequence them, and basically read the barcodes.
Let me make sure I'm wrapping my head around this. You're saying we are literally attaching a microscopic barcode to a drug, firing it into a mouse, and then scanning the surviving cancer cells like items at a grocery checkout.
That's exactly what they're doing. They know precisely which molecular weapon failed and which one worked, cell by individual cell.
That is absurd.
And the best part, they aren't keeping this data isolated in single labs.
Right, the data is being compiled globally. The sources mention massive research consortiums like PDXNet in the United States and EuroPDX in Europe, building these global biobanks.
Right. They compile all of this multi-omics data, the drug responses, the genomic sequencing, and they feed it all into massive machine learning networks to train AI to predict drug responses based on genetic profiles.
Okay, zooming out for a second to recap this. We are speeding up the avatar process using transparent zebrafish to literally watch glowing cancer cells react to drugs in real time.
Yes.
We are using CRISPR to selectively edit genes inside the living host to find weak points. We are reading single-cell data and literally barcoding nanoparticles to track drug effectiveness, all while feeding this massive amount of data into global AI databases.
It's a total technological revolution.
We're essentially creating a biological search engine. Think about that. What if a doctor could query a global database of thousands of living avatars to instantly find the exact molecular match to your profile?
Which brings us to the ultimate question, doesn't it? How is this translating to the clinic?
Right, because all this tech is cool, but what does this all mean for the patient? How is this technology actually saving human lives today?
Let's look at some real-world clinical applications, starting with targeted therapies. PDX models are unearthing completely out-of-the-box solutions that we'd never find otherwise.
Like what? Give us an example.
For instance, appendiceal cancer, which is cancer of the appendix, often secretes this incredibly thick, abundant extracellular mucus.
Mucus. Gross, but okay.
Yeah, it's essentially slime. And this mucus acts like a physical field, completely blocking chemotherapy drugs from ever reaching the cancer cells underneath.
So the chemo drug just bounces off the slime. It never even touches the tumor.
Basically, yes. But by utilizing PDX models, researchers tested a totally novel combination of bromelain and N-acetylcysteine.
Bromelain?
Yes, bromelain is actually an enzyme naturally found in pineapples.
Wait, pineapples?
Together, these act as powerful mucus-busting agents. By giving the avatars these agents first, they successfully dissolve the protein bonds in the mucus shield, allowing the standard chemotherapy to finally penetrate and destroy the tumor.
That's brilliant, using a pineapple enzyme to dissolve the armor. But what about when cancer mutates to resist a targeted drug? Because that's a huge problem.
Oh, it is. And PDX is vital for predicting those resistance mechanisms. Take colorectal cancers, for example. We often treat them with EGFR inhibitors.
EGFR inhibitors.
Right. Think of EGFR as a cellular antenna that receives signals telling the cancer cell to grow and divide. If we inhibit it, we jam the antenna and the tumor shrinks. But eventually, the tumors almost always mutate and become resistant.
They figure out a workaround.
Exactly. By continuously studying colorectal PDX models, scientists discovered exactly how the resistance happens. The surviving cancer cells suddenly spike the activity of completely different cellular antennas, specifically ones called HR2 and HR3.
Ah, so they just build a detour to bypass the blocked EGFR pathway?
That's exactly it. And because the PDX avatar revealed this specific detour pathway before it happened in the human patient, doctors now know to preemptively strike.
Oh, so they can block the detour before the cancer even takes it?
Exactly. They could administer pan-EGFR antibodies or a combination of HR2 targeted therapies before the cancer even has a chance to escape.
It's predictive oncology. You're anticipating their next move.
Yes. And it's even more impactful in immunotherapy optimization. Earlier, we used the analogy of the cancer acting like a hacker, exhausting the immune system through trogocytosis.
Right, the nibbling, stealing the armor.
Well, in humanized PDX models, researchers recently discovered a highly specific subset of T cells that they officially named EBO, which stands for burned out CD8 T cells.
Burned out T cells.
Yeah, these cells multiply rapidly in the tumor microenvironment, but they are completely dysfunctional. They just take up space, they don't fight, and they are a major reason why some patients resist immunotherapy.
So they're just dead weight.
Pretty much.
Pretty.
But by testing various compounds in these avatars, researchers found that administering an existing drug called evolumab actually prevents these burned out cells from expanding in the first place.
Wow. And then there's CRT cell therapy, right? We mentioned upgrading the immune cells like software. And the PDX models are the ultimate testing ground for this.
They absolutely are.
Yeah.
CAR-T cells are human T cells engineered in a lab to aggressively attack cancer. But the danger is they sometimes mistake healthy tissue for cancer, causing severe, sometimes fatal side effects.
Friendly fire.
Exactly. So researchers used PDX avatars to test next-generation CAR-T cells engineered with what are called synapse receptors.
Synapse. How does that mechanism work?
Think of it like a logical AND gate in computer programming.
Okay.
The CAR-T cell requires multiple specific antigen keys to activate. It has to recognize antigen A and antigen B simultaneously to turn its killing mechanism on.
I see.
If it only sees antigen A, which might be present on a healthy cell, it stays totally dormant. It only attacks when it finds the specific, complex combination unique to the tumor.
That is literally programmable immunity.
It is. It's like writing code for your white blood cells. And they are also testing CAR-T cells in these avatars that are engineered to secrete specific chemical signals, like IL-17 and a chemokine called CCL19.
And what do those do?
These signals aggressively alter the tumor microenvironment, effectively calling in other immune cells and shutting down the cancer's defense mechanisms from the inside out.
So if we look at the clinical triumphs here, PDX models are being used to break down physical barriers like mucus shields in appendiceal cancer and to predict mutational detours in colorectal cancer.
Yes.
They are identifying entirely new subsets of burned-out immune cells and serving as the proving ground for futuristic, programmable CAR T cells that require multiple genetic keys to unlock their cancer-killing potential. We aren't just reacting to cancer anymore. We are running highly customized preemptive simulations. Are we witnessing the end of trial and error medicine?
I truly believe we are. The sheer fidelity of these models is transforming oncology from an observational science where we wait to see if a drug works into a predictive science where we know it will work before we administer it.
So to summarize our journey through the sources today, we started with the foundational breakthrough of Rygaard and Paulson's first nude mouse in 1969.
A long time ago.
Yeah. We explored how humanized immune avatars recreate the complex battlefield between T cells and tumors. We acknowledged the philosophical and biological flaws of the matrix, like stroma replacement, the ship of Theseus, and subclonal survival of the fittest.
Right.
But then we saw how transparent, glowing zebrafish, CRISPR gene editing, and DNA barcoded nanoparticles are supercharging our ability to gather data at the single-cell level.
And this raises an important, really provocative question for the future of medicine.
What's that?
Well, as we begin using patient-derived extracellular vesicles and relying on global machine learning networks to map out every conceivable cancer mutation across thousands of these living models, we are moving beyond just animal testing. We are essentially building biological and digital twins of ourselves.
Biological twins, wow. And that brings us right back to the architect we imagined at the beginning of the show. We no longer have to build the skyscraper just to see if it will fall down in a storm.
You have the simulation.
Exactly. If we can perfectly simulate a disease and its cure in an avatar living safely outside of your body, how far are we from a future where a patient never has to experience the toxic, heartbreaking side effects of a failed treatment ever again? Think about that incredible possibility the next time you hear the word diagnosis. Until next time.